evening, everyone. This is Shelley Harshberger, and I'm here with our to review. And tonight's webinar is going to be our all-in-one biology. This will cover TEAS 7 science subjects, S2.1 through 2.5. Before we dive into that content, I did want to go over all of the amazing things that we have available to you to help you prepare for the TEAS. As you can see on this screen, we have our free live webinars. I have scientific reasoning coming up next month. We've got an anatomy and physiology in between there. And I believe we also have a chemistry. So you can access all of those by that top QR code. You also have the ability to re-watch old webinars by that second QR code. These are recorded nights like tonight that you can take a look at. Also, remember that we have our question bank. Our question bank is over 1,500 questions that you can go through. You can take the webinar question, or not webinar, sorry. Take a practice test with these questions, and you can get the rationale as to why the answer is correct and why the other answers are incorrect. And this is available to you as a free trial. We also have on-demand videos. So tonight is an overview. It gives you a lot of information about these topics on a somewhat superficial level, and then the on-demand videos take you deeper. So tonight you can get a feel for where your strengths are and where you might need a little assistance, and then those on-demand videos could help you hone in on those. We also have private tutoring available. I did several tutoring sessions today, and so that QR code is there as well. And then we have two packages, the SurePass and the SurePass Pro, where if you bundle, you can save a little bit. And so let's take a look at some of those in a little bit more detail. So the SurePass helps you bundle the 1,500 questions, helps you track your performance, and it will have targeted videos and objectives. So when you take the practice test, if you have a question that you're struggling with, it will actually take you straight to the on-demand video where you can learn about the particular topic. Then the SurePass Pro has the same general idea, but it also adds private tutoring, which is really cool as well. Like I said, I had several tutoring sessions today. If you are a social media follower, we are everywhere. We are on Instagram, we are on TikTok, we are on Facebook, and you can always reach us via email as well, prenursing@archerreview.com. These are the tutors. So my colleague Tammy is our English and reading specialist. I am your science person, and Mr. Brandon Kraft is your math expert. So you can also scan those QR codes to reach out and schedule a tutoring link. So tonight, as I said, we are going to cover the TEAS science, the biology section. So that's going to be things like macromolecules, cells and microorganisms. We'll look at genetic material and proteins. And we'll also look at genetics, which is the Mendel's laws of inheritance, things like Punnett squares. So kicking right off with macromolecules, our learning objectives here are to really make sure we understand what macromolecules are, recognize their structure, and then identify their function. So macromolecules are considered polymers. We could use those two words synonymously, and they are made up of smaller components known as monomers. So let the prefix of those words help you figure out what they mean. So poly, that's the prefix at the beginning here, poly, that means many. The prefix here, mono, means one. So it's saying that this is a larger molecule made up of many individual or singular units. Macromolecules make up us, and macromolecules are also acquired from our food. So carbohydrates, proteins, things like that we get from our food. And they are broken into the monomers, the individual components, in order to get our body's fuel. And that fuel is ATP. That is what is pictured right here. So it's showing us that if we ate an apple, that apple would be broken down in our body to our body's fuel, or better known as ATP. Oh goodness, I was trying a new writing feature and I don't want it all over our screen all night. That's not good. Let's go ahead and erase that. I thought that new writing feature was going to be pretty great, but not if it was going to stay on every screen. My goodness. All right, here we go. So the macromolecules, as I said, there are most of them are required to our food, but there are four primary macromolecules. There are carbohydrates, lipids, nucleic acids, and proteins. And we will take a look at all four of those tonight. So when we first look at carbohydrates, the monomers of carbohydrates are monosaccharides. And so mono, again, remember, means singular. And then saccharide is the word that stands for carbohydrate. So I want you to just kind of tie the word carbohydrates, sugar, or saccharide in your brain almost as synonyms. Anytime you hear saccharide, either think carbohydrate or sugar. Then when those monomer units are put together, they form that poly, remember that word means many, polysaccharide. Okay, and you can see down in our visual here, this is one monomer linked to another monomer linked to another, and so on to create our polysaccharide. Now, an example of a polysaccharide would be glycogen that is stored in our liver until we need that fuel. Okay, and so that would be an example of that. So carbohydrates are recognized as monosaccharides, meaning that it is just built of one saccharide. An example of that being glucose. Glucose is a single saccharide sugar. There are disaccharides. That di in front of there means two, means the number two. And an example of that would be sucrose. Then remember that prefix poly in front of there means many. And an example of that, as I said earlier, would be glycogen. And glycogen can be stored in our liver, as I said earlier, but it can also be stored in our muscles until needed for energy. And then that glycogen is going to be converted back to glucose, where our body then can take that energy in the glucose and convert it to ATP, our body's fuel. Lipids, another macromolecule. The monomer of a lipid is a fatty acid, and that's what we see in this structure here. An example of a polymer of a lipid is the lipid bilayer of our cell, a lipid bilayer of our cell membrane. That's the outer container of our cell membrane. That's what keeps these substances within our cell, and it also helps monitor what enters and exits our cell. And so that's an example of a lipid, and we're going to focus on that a little bit more. The cell membrane is a really important component to biology and to understanding cellular function. So this is a picture of our cell membrane, and what we see on either side of it is a watery environment, okay? Because as you probably already know, our cells have a lot of water inside of them. They're primarily water, and our bodies are primarily water. And so our cells are constantly exposed to a watery environment. Well, lipids are funny. Lipids have areas that prefer water and areas that don't prefer water. And so that's what we're going to look at a little bit more closely. But this is considered a lipid bilayer. Now, again, let those prefixes help you. If the word bi is in front of a word, that generally means two. And so what this is telling us is that the cell membrane is a layer of two lipid rows. So we see this is one layer of lipids, And this is another layer of lipids. And that's where we get lipid bi or two layers. Oops. We want to go back. I had other things on there that I want us to be able to see. So they orient special because, as I said, they have unique features regarding water. So the tails are what we call hydrophobic. Now, again, we're going to let those words help us out here. That prefix hydro means water. Think of what you know about the word phobic. If you have a phobia, it means you're kind of afraid of. And so we can use that to help us remember that these are water hating. Okay, we can use that word phobia as like fear or hating to say that this part of the lipid is not fond of water. It does not want to orient towards water. Okay, and the reason that is its chemical structure. It's what's referred to as non-polar, and water is polar, and those two don't get along, okay? And so the tails of the lipid will orient towards the inside of the cell membrane to get away from the water. And that's what we see right here. These are hydrophobic tails, and they're orienting towards the inside to avoid this water. They don't like the water, they don't want any part of it, and they're avoiding it. Now, the heads, my goodness, the heads, on the other hand, are more water loving, okay? See that word hydrophilic. This means that they love the water. I'm trying to draw a heart with my cursor, so bear with me that my drawing skills aren't the best. So they love water. And that's why those heads are oriented towards the water. They think that's a good thing, okay? And that is really important as you get deeper into your biology studies and into your studies as nursing. You'll understand that substances will be able to kind of enter and exit between these molecules based on their orientation and based on their charge. And so the structure of the cell membrane becomes very important in that context. All right, we're going to move right on. And somebody's telling me that Philadelphia is kind of like that. I respect that. I understand that. All right, so our next macromolecule that we want to look at is protein. And protein's monomer is an amino acid. And there are probably 4 important things to know about the structure of an amino acid, something that would certainly be on the TEAS. One of the important things to know is that they are composed of an amine group. That's what's right here, a nitrogen with hydrogen. They're also composed of a carboxylic group, a carbon with 2 oxygens and a hydrogen. They have a central carbon right here. And then remember that there are 20 different amino acids. And what distinguishes one amino acid from the other is this R side chain right here. That varies depending on which of the 20 amino acids it is. Now, the protein polymer, when these amino acids link together, that's referred to as a poly, many peptide. So again, we have all these synonyms in science. You want to, when you see that word peptide, think protein. Protein peptide, put those 2 words together in your brain. And so an example of protein would be like a muscle protein. That would be an example of a polypeptide all folded up and in a structural unit of something that we can kind of wrap our head around. So one of the important ways that proteins work within our bodies, ways that you would certainly need to know for the TEAS, is that they assist chemical reactions without being consumed. And they do this in the form of what is known as an enzyme. And how they do this is that they lower the energy required for that reaction to occur. And they connect with whatever substance that they need to connect with that's referred to as a substrate in this kind of lock and key fashion. So notice how in the middle here, it looks like a key fitting into a lock. That's how they interact with the substance that's referred to as a substrate to lower that energy required for the reaction to occur. Now, what's important to be aware of, though, with enzymes is that they're very sensitive to temperature changes and pH changes. And so they can break down and their shape change if they're exposed to the wrong temperature or the wrong pH. And that will impact their function. And then they won't be able to function and do the job of decreasing the energy needed for that reaction. Now, our final one that we want to talk about is nucleic acids. The monomer of nucleic acids is the nucleotide. And the nucleotide has three primary parts. This phosphate group right here, the sugar right here, and then a nitrogenous base. And the nitrogenous base, much like we said with the amino groups and the R side chain, it's this base right here that determines which nucleotide it is. Whether it's adenine, thymine, guanine, or cytosine, all relies on this base right here. And this base, this nucleotide then, makes up one little chunk right here of the DNA. There's our base, and then our sugar and phosphate go along the sides there. Now, RNA, we would have pretty much the same setup, though the sugar would be a different sugar, and it would also just fit like one spot of it right there. Now, when DNA coils all up into a chromosome, that's what we see here as our example. And that DNA would just coil up really tightly in here, and it would be wrapped with histone protein. And those histone proteins then would protect that chromosome. All right, so some things to be aware of since we have two versions of nucleic acids that we focused our attention on. We've got DNA and RNA. The things that they have in common is that they are both involved in protein synthesis, and they can be impacted by changes. Well, DNA can be impacted by changes referred to as mutations, and RNA can be impacted by alterations. I wouldn't necessarily call that a mutation. That's something that I'll want to update in the future. Now, what makes them different is that DNA, as we see here, is double-stranded, whereas RNA, as we see here, is single-stranded. DNA, when we pair those bases to create the steps in our ladder, A pairs with T, adenine pairs with thymine, and cytosine pairs with guanine. Whereas when we use RNA, whether it's in when it's in transcription, A is going to pair with U, so adenine is going to pair with uracil. There's no thymine in RNA, it's uracil. And then cytosine is still going to pair with guanine. So DNA, its whole entire purpose is to provide the recipe for life. And then that recipe is read in chunks referred to as genes, and then those genes direct the construction of protein. RNA gets involved in that gene to protein transfer. RNA helps us make that come true because the instructions for the proteins are in the nucleus on the DNA, and RNA gets them out to the ribosome where they are built. And so there are three types of RNA that are involved in that process, and we will look at those three types when we get to protein synthesis. So we'll look at them more closely later, but the first one is messenger RNA, the second one is transfer RNA, and then the other one is ribosomal RNA. All right, so we've got some key takeaways for this segment. Carbohydrates come in many forms. Mono, singular, di, two carbohydrates together. Poly, essentially more than two. It could be 3 to hundreds. And they're going to play a key role. They're going to actually help us with cell recognition for our cells to recognize themselves as self and not attack, as in like an autoimmune issue. And they're also, most important in energy provision. So carbohydrates are the primary source of where we get the energy to function. Lipids, as we saw, most importantly, create that lipid bilayer, that semi-permeable barrier that regulates what moves in and out of a cell. Proteins, as we saw, they're made of amino acids. They play a very big role as enzymes. And then, excuse me, and then enzymes function to support chemical reactions to make it to where there's not as much energy needed for those chemical reactions. Remember that nucleic acids in the form of DNA holds our genetic code. RNA is going to be important in protein synthesis in the role of transcribing and translating. And then we didn't talk much about this, but ATP, our energy's fuel, is also a nucleic acid. It's a slightly modified one. It has 2 extra phosphates, but it is also a nucleic acid. All right, so now we've got a couple questions. I do want to tell you before I dive into the question that if you are on Zoom, only I can see your answers. So you don't have to feel nervous or insecure that people are going to see a wrong answer. I don't anticipate any wrong answers, but just so you got the confidence to put your answers out there. And please do. That's one way to make it more interactive. If you are on our other streaming services, YouTube, Facebook, and so on, I'm sorry. I will be able to see, everyone will be able to see those, but that's okay, because I think you'll have the right answer anyway. All right, which of the following best describes the characteristics of enzymes? A, they are consumed during chemical reactions and increase the energy required for reactions. B, they assist chemical reactions without being consumed and lower the energy required for reactions. C, they connect with substrates randomly and are unaffected by temperature and pH. And D, they speed up reactions but are not specific to the substrates they interact with. Now, I always give us 45 seconds for these. So I'm going to get my timer going. Take your time. It is pretty common that most multiple choice questions on any form of exam, you get about 35 to 45 seconds. And so that's always kind of my setup. So here we go. We'll be back in touch in 45 seconds. I'm seeing some correct answers out there already. All right, I think most of you had this. Our correct answer is B. They assist chemical reactions without being consumed and lower the energy required for the reactions. That is correct. What makes A incorrect is that they are not consumed during chemical reactions. Enzymes can be used over and over and over. And also, they decrease the energy required for those reactions. They don't increase it. So that's what makes A incorrect. C, the fact that they connect with substrates, it says randomly, no. Remember, it's like a lock and key fashion. They are very specific to their enzymes. And so that's what makes that one incorrect. And then that also helps explain D. They are specific to their unique enzyme. The enzyme is specific to its unique substrate. And so that's what makes that one incorrect as well. All right, let's do another. Which of the following statements correctly differentiates DNA from RNA? A, DNA contains the base thymine, while RNA contains uracil instead. B, DNA is single-stranded, while RNA is double-stranded, and both use AT base pairing. C, both DNA and RNA are responsible for transporting amino acids during protein synthesis. Or D, RNA is the recipe of life, while DNA assists in translating and transporting genetic code. You got 45 seconds. I'm seeing some correct answers out there. Okay, if you chose A, you would be correct. So DNA has thymine, whereas RNA has uracil. Now let's break down why the others are incorrect. B, DNA is single-stranded. No, it is double-stranded. So they flipped those, okay? And then also they don't both use A and T base pairings, right? Only DNA does. Both DNA and RNA are responsible for transporting amino acids during protein synthesis. No, because DNA is like in jail in the nucleus. It is locked into that nucleus. And so it can't do any transportation of amino acids during protein synthesis because that occurs on the ribosome outside of the nucleus. And then D, it just switched the two. DNA. If this said DNA and if this said RNA, then that would be correct. But since it didn't, that makes that an incorrect answer. Okay, our next topic is focused on cells and microorganisms. So in this one, we are going to look at how cells are classified. We're going to look at the structure and function of the key organelles inside our cells. And then we're also going to identify some common microbes that cause disease, known as pathogens. So I really like this video or this visual. It really helps break down the different types of cells that exist in our world. So we have two broad categories of cells. We have eukaryotic cells, eukaryotes, and we have prokaryotes. Prokaryotes, that is where a lot of the microorganisms that we'll talk about towards the end of this section come in. In this case, they're single-celled, uni means one, and they're very simple. They have free-floating DNA, meaning it's not locked in a nucleus. And they really don't have what they refer to as true organelles. They have ribosomes. That's what those little round balls are in there. But that's it. They're very basic. They consider them primitive cells. The cells that make up us, make up organisms like us, are multicellular, meaning many cells. They're very complex. They have a nucleus. The DNA is protected inside that nucleus. And they have what are referred to as membrane-bound organelles, meaning the organelles within the cell have membranes around them. Now, eukaryotes break up into two types. We've got our animal cells right here, and we've got our plant cells. And plant cells have a rigid cell wall. That's this big green structure all around this visual, whereas we do not have a cell wall. We have a cell membrane, which plant cells also have, but we do not have a wall. We have a mitochondria. That's what we see right here. Plants have a mitochondria and chloroplast. So plants actually have two ways to convert energy. We only have one. We have lysosomes. They lack lysosomes and they lack centrioles, two things that we have. And then we have many small vacuoles within our cells to store water and other substances, where they have this one large vacuole right there. All right, now obviously, because we're focused on the TEAS, and the TEAS knows that your future is nursing, the focus from this point forward, now that we understand how cells are classified, is on eukaryotic animal cells. These are the cells that make up you and I. And so we're going to look at the structures that are key and important in those animal cells, or that form the animal cells. So for example, the first one we talked about already, we talked about that cell membrane. Notice that this lipid bilayer right here should look very familiar to you, okay? And what the job of the cell membrane is to enclose the cell. And it's also very selective. It selects what passes through. This is what I talked about earlier when I was talking about lipids, that some molecules will be able to just kind of squeeze right through these lipid molecules. Others will need proteins that are within that cell membrane to assist them, but some of them can just squeeze right through. And then the other function is that they separate the internal cellular environment from the external cellular environment, and that's important as well. Cytoplasm is another important component of a cell. It is a water-based jelly-like fluid that all of our organelles are suspended in. It is water-based so that chemical reactions can occur within that jelly-like substance. It also aids in supporting the structure of the cell and helps hold the organelles in place, but it's fluid. They can move around. Then we have the Golgi apparatus. That's the orange folded, it looks like folded sheet. Its function is to process proteins. So when we look at protein synthesis later, we look at how proteins are built. And then once we have a big long chain, a long polypeptide, that chain will go to the Golgi apparatus or the endoplasmic reticulum to get folded into its functional shape. They also process lipids into little round vesicles, little round looking, almost looks like balls, to transport those lipids in and out of the cell. Lysosomes are also in our cells. These are what look kind of like oranges in our visual here. They are responsible for breaking down waste and denatured proteins. So we talked earlier about how enzymes could be impacted by pH and temperature. So if that enzyme is impacted, it breaks down, or what is referred to as denatured, and that protein then would be sent to the lysosome to be broken down. Mitochondria. Mitochondria is the star of the show when it comes to energy transformation. It is going to take the energy that is in our food and transform it into our body's fuel, ATP. ATP is like our body's gasoline, and that process is called cellular respiration. The nucleus, as I had said earlier, is where our DNA lives, where it resides. It cannot leave the nucleus, and the nucleus is an added protection for our DNA. So it's going to hold that genetic material, and in some ways, it's going to kind of be like the brain of the cell. It's the overseer, the boss. It controls the actions of the cell. Now, I want you to really pay close attention to the nucleolus. I feel like this is an easy thing to confuse. Notice this word has Leo in the middle of it. So just like it has the word Leo in the middle of it, the nucleolus is in the middle of the nucleus, okay? And its job is to produce and build ribosomes. That is its sole job, produce and build those ribosomes for us. Now, the smooth endoplasmic reticulum is right next to the nucleus. We see that it's also like folded sheets. And we've got a rough endoplasmic reticulum that we'll look at in a minute. The only difference is that there are ribosomes on the rough endoplasmic reticulum, or the only visual difference. The smooth endoplasmic reticulum is responsible for making lipids. Remember, lipids are like our fats, and hormones. And they also have a very unique role in detoxifying our body from drugs and other toxins. Now, the rough endoplasmic reticulum, I said visually the only difference was that there were ribosomes on the rough endoplasmic reticulum, so it looks rough, and that's where it gets its name. Now, its job is to assist with that protein synthesis because of the ribosomes, but it also does that folding of the protein that I talked about earlier, as well as the Golgi apparatus. And so we can see that the rough endoplasmic reticulum is going to help fold, and then the ribosomes on it are going to be what helps synthesize or make those proteins. Vacuoles. Vacuoles are kind of like our cell's garbage bags. It's going to store fluids, it's going to store carbs, it's going to store some toxins. Most of them are stored for expulsion, to be get rid of. Sometimes they can be used for other things, but most of the time it's just used to store and then take them out, take the trash out, so to speak, later. All right, the next topic we're moving on to is microorganisms. So while we looked at the cells that make up us, we also talked about how there are prokaryotic cells. Those are very simple cells, but there are also some microorganisms that are eukaryotic as well. And our focus is on the ones that cause disease. So the microorganisms that cause disease are known as pathogenic microorganisms. And that is our next focus. So there are 5 broad categories of microbes that we're going to focus on. We're going to focus on the category of microbes that are just referred to as microscopic animals. We'll focus on protozoans, fungi, bacteria, and while not technically a microbe, and we'll get to this, viruses. And viruses kind of get thrown into the microbe category, basically because where else when we put them? They do cause disease, but they have characteristics that actually make them not 100% a microbe, and we'll get there. So microscopic animals, the broad category that we'll really focus on here is helminths. These are parasitic worms, things like tapeworms, roundworms. They can live in our intestines and cause malnutrition. They can obstruct our intestines and make it impossible for us to get rid of waste. If you have pets, most of us have pets, when your vet asks you to bring in a stool sample, a poo sample, this is what they're looking for. They literally take some of your pet's poo, mix it with a fluid, and look at it under the microscope to see if they can spot any worms. Protozoans. Now, protozoans fit in the category of protists as well. These 2 words are synonyms. Protozoans, protists, synonyms. They are single-celled eukaryotes, so their cells are complex like ours, but they're only made of 1 cell. They're actually the largest group of organisms on the planet. Very creepy, but true. And the primary example is the disease they cause is malaria. And malaria is actually caused by the Plasmodium species. And what this does is it infects mosquitoes. And then when the mosquito goes to bite you and draw your blood, it actually injects a little bit of numbing agent so you don't feel while it's pulling the blood out. And within that numbing agent is where it infects you with this single-celled eukaryote. And then that will go to our liver and invade our red blood cells and cause all the bad symptoms of malaria. So be cautious with this visual, though, that this visual is not telling us that protozoans are mosquitoes. It's showing us that that's how you could get infected by a protozoan. So be attentive to that. Fungi. Now, fungi is a broad category. Obviously, not all of them are microscopic. We can see mold, we can see mushrooms. We could see, well, I don't know if we really can see yeast unless we see the effects of it in bread. So not all are microscopic versions, but there are microscopic versions as well. They are eukaryotic, so that would be a complex cell like ours. In the nature sense, they're decomposers. But in the sense that we're worried about as microorganisms, they're parasites, okay? And in this case, they cause diseases such as athlete's foot, or notice this, be attentive to this, ringworm. Is fungi a worm? No, it's not a worm. But that's the name of this condition right here. And it got its name because it looks like a worm underneath the skin. but it is not a worm. So make sure you realize that ringworm is a fungus, okay? It is not a worm. That is something that can catch you, that you can have issues with, so be attentive to that. Bacteria. Now, bacteria is a prokaryote. Remember, prokaryotes are those very simple cells, right? They have DNA that is not enclosed in a nucleus. They don't have any true organelles. They're very, very simple. Now, what will be very important to you as you go through your nursing career is that they are classified by their type of cell wall. So if they have a thick cell wall, they're gram positive. So that's what we would see right here in the purple. If they have a thin cell wall, they are gram negative, and that's what we would see right here in the pink. So purple, gram positive, pink, gram negative. Okay? And that is important because many of the antibiotics target the cell wall. So depending on the thickness of the cell wall, that will determine what the doctor prescribes. It is also important to note that not all bacteria are pathogenic. The bacteria that is in yogurt, lactobacillus, that is actually very beneficial. We have bacteria that lives in our digestive system, and that is incredibly beneficial to help us break down foods our body cannot break down. But the pathogenic bacteria that's out there could cause things like food poisoning, meningitis, sepsis. Remember that sepsis is like blood poisoning, meningitis is a poisoning of the layers around our brain. So there are plenty of bad bacteria as well, or pathogenic bacteria as well. Now, finally, on to viruses. This is where I said they're in this category because where else will we put them? But they actually call them non-cellular entities because they cannot reproduce on their own. One of the characteristics of life is the ability to reproduce. And because viruses cannot reproduce on their own, they're not actually classified as like a living organism. And so they have to call them something, and they refer to them as non-cellular entities. They're very unique in that they will either have DNA or RNA, and it's just enclosed in a protein coat. Viruses that we are familiar with are things like the common cold, measles and mumps. Those are vaccines that you get for measles and mumps generally in elementary school or before elementary school. And then, of course, we know COVID-19 was a virus as well. So let's take a look at our key takeaways for this section. Cells are going to be classified as either prokaryotic or eukaryotic based on their complexity. Remember, prokaryotic is very simple, eukaryotic very complex. Eukaryotic animal cells, the cells that make up us, have key organelles. The Golgi apparatus is for protein and lipid processing. The mitochondria is going to transform energy. The nucleus is going to manage our cellular functions and hold our DNA. The nucleolus, remember Leo is inside of the nucleus, is going to help build ribosomes. We've got our smooth ER that is critical in detoxifying us, and we've got our rough ER for protein processing. Then when we look at pathogenic or infectious microbes, we've got microscopic animals such as parasitic worms. We've got protozoan or protists, those are single-celled eukaryotes, for example, the one that causes malaria. We've got fungi, a parasitic form would be ringworm. We've got bacteria that are prokaryotic, and they can be gram positive with a thick cell wall or gram negative with a thin cell wall. And then we got those viruses that are non-cellular entities because they cannot reproduce on their own. All right, we've got a couple of questions. Let's see what we've learned so far. Oh, this one, I forgot about this one. So this one matches. So I am going to give you the 45 seconds and you just kind of process in your brain what goes with what. and then we will reveal them after the 45 seconds. I think you got this. Good luck. Okay, let's take a look. So our first one, ribosome, makes protein. Our next one, rough ER, processes protein. Nucleus controls the cell. Vacuole for waste storage. Remember I said those were like our trash bags? And then, of course, smooth ER makes lipids and hormones. Let's do another. Ringworm is actually a fungus, protist, virus, or microscopic animal. Get those answers out there. I'm seeing some correct answers already. You guys rock. Okay, I think almost everybody got this correct. It is actually a fungus. Remember, it is not a worm, despite the name. Remember that protists are things like malaria. Those are single-celled eukaryotes. Viruses are non-cellular entities. And microscopic animals are things like parasitic worms, like the worms that your veterinarian would be checking your dog or cat's stool for. All right, our next topic. We are about halfway through this evening. Thanks for sticking on with me. Is cellular processes. In this category, we are going to look for the key structures and functions in DNA replication and cellular division. We're going to look at the relationship between the nucleus, chromosomes, genes, and DNA. And then we're also going to look at the difference between mitosis, which is somatic cell division, and gametic cell division, which is meiosis. And we'll even make sure we understand what those two words mean. Somatic is body cell, gametic is sex cell, but we'll get to that. So one thing that's important to be aware of is that a cell has a life, kind of like we do. It has stages. So like we have newborn, toddler, teenager, senior citizen, and so on. The cell also has its stages of life. Its longest stage of life is interphase. And that's what we see in our visual right here. So this whole time here is interphase. And within interphase, during G1, during this time right here, it's going to do its normal cellular function. If it's a heart cell, it's going to do heart cell functions. If it's a muscle cell, it's going to contract and help you move, and so on. During the S phase, DNA is going to replicate. It's going to double so that the cell can divide. During the G2 phase right here, it's going to do its final preparations for division. And division is going to occur in this process right here, known as mitosis. And this is where ultimately it separates that doubled DNA. Remember, DNA replication occurred right here, and it doubled that DNA. Well, now in mitosis, it's going to go through the process of splitting that DNA into two separate cells. Those cells will finally split right here into two new cells, right here. Cytokinesis. That's the final step of a cell's life where it goes into 2 new daughter cells. And we're going to take a look at each of these then more closely. So our first focus is DNA replication, where that DNA is doubling, okay? But to get to DNA replication, we've got to do a little bit of DNA background. So remember that the nucleus is where DNA is hanging out. Remember that DNA is what they refer to as a double helix. It means that it's got 2 sides to it, okay? The rungs or the steps of the ladder are those nitrogenous bases. And that's where adenine is pairing with thymine and guanine is pairing with cytosine, okay? Now, remember that they are made of monomers, and that monomer is a nucleotide. That's what we see right here. And that nucleotide is made of a phosphate group, a sugar, and then there's the specific base. That's what makes that cytosine. If we were looking at this one, we'd still have a phosphate, we'd still have a sugar, but the base is different, and that's what makes it adenine, okay? And it's important to know those structures in order to make sense of DNA replication. So the first step in DNA replication is an enzyme unwinding DNA. This enzyme is DNA helicase, and it is going to unwind, as we see in the visual right here, the DNA and separate those bases. Those bases are connected by very weak bonds. Those weak bonds are called hydrogen bonds. And so helicase is just going to go up through here and break those bonds to separate the DNA. With the double-stranded DNA unzipped, it's going to serve as a template for 2 new strands. So new matching bases, A to T, C to G, are going to be added to form 2 identical DNA molecules. And that's important to understand. We've got 2 identical DNA molecules. And each one's going to have an old strand, and each one's going to have a new strand. And that's what we're seeing right here. See how this was our original DNA, and we've got one of the original strands that's going to get new stuff built on it, and we've got the other original strand that's getting new DNA built on it, okay? That ensures that both new cells get the exact same genetic information. So remember we said DNA replication takes place right here. Once that's done, the cell's going to continue to prepare right here for this process right here, mitosis. And that's what we're going to look at next. So there are 4 stages to mitosis: prophase, metaphase, anaphase, telophase. Now I know in like the high schools, they generally try to use like an acronym or something to remember that. They'll say PMAT, or they'll do PMAT-C because cytokinesis is where they actually split. Whatever works for you, those are the order that they are in. I know that there sometimes exams like TEAS can ask for something like that. So that might be an acronym that can help you remember the order of them. So prophase, what's going on in prophase? Well, our chromatin is another name for our DNA when it's just kind of hanging out in our nucleus. It's going to condense, though, in prophase into chromosomes. So I think that's an important distinction to think about. DNA, when a cell is not preparing to divide, doesn't sit around in chromosomes. That's only when it's getting ready to go through mitosis. So then when it's getting ready to go through mitosis, or meiosis for that matter, it will condense into chromosomes. And the chromosomes consist of what they call 2 identical sister chromatids. So this is a chromatid, this is a chromatid. okay? And they're joined in the center by this, which is known as a centromere. So they're two identical sister chromatids with a centromere in the center. That's during prophase. During metaphase, then, those chromosomes are going to line up in the center of the cell. That's what we see right here. And they're going to be equal from the opposite ends of the cell. If you remember looking at anything under the microscope in maybe biology class in high school, sometimes you would look at onion root tips, and metaphase would always be the phase that was the easiest to see. Now, anaphase, this is where the cell is starting to pull apart that double DNA. Remember, we had identical sister chromatids. Well, one of those sister chromatids is going to go into a new cell, and the other sister chromatid is going to go into a new cell. And that's what we see happening here. See how they're pulling them apart? There's a chromatid, it's being pulled apart. Then in telophase, they're completely separate. They've reached their opposite ends. We start getting our nuclear membrane that will be the nucleus, the membrane around the nucleus. Our chromosomes start breaking down into chromatin again, and the spindle fibers that help pull them apart will just kind of degrade. And we see at the end of telophase, we have nearly two new cells, but they have not fully split yet. They fully split in cytokinesis. Cytokinesis is what fully separates them into two genetically identical daughter cells. And so those identical daughter cells are going to have the same number of chromosomes as the original parent, the same DNA as the original parent, and for that matter, the same DNA as each other. Now we're going to look at meiosis, okay? The division we just got done looking at is what every single body cell in our body goes through except for the sex cell, except for the sperm and egg cells. So meiosis divides what they call germ cells. Now, if you're not familiar with some of the English language, don't mess this up. Germ cells in this context does not mean like, oh, you're going to get germs on your hand. Germ cells in this context means sex cells. The spermatocytes, the precursors to sperm, and the oocytes, the precursors to eggs. Okay. Now, what's unique about meiosis is that we've got to create some genetic diversity. We don't want to be identical to our parents. We don't want to be identical to our siblings. And so genes have got to be shuffled in some way, shape, or form so that we don't end up being identical to our siblings. This occurs through a process known as crossing over. Okay, and we'll look at what specific phase that occurs in. That is often a TEAS question. Now, we have a goal here, though. When we're making a sperm cell or an egg cell, our goal is not our 46 chromosomes that we have in every single body cell. Our goal is 23 chromosomes. And the thought here might be, well, wait a minute, why? Why do we need half? Well, let's think about this for a minute. If we had, this is a sperm right here. If that sperm had 46 chromosomes, and if this egg had 46 chromosomes, and they united, how many chromosomes would our baby have? 92. Well, that's not right, right? And what if that baby then had an egg with 92 and so on, right? So we can't do that. That does not work. We can't have the same number of chromosomes as our body cells because when that sperm and egg unite, we have to have the right number. And so one of the primary goals of meiosis is to get us to the correct chromosomal number. So if we have 23 chromosomes in our sperm and we have 23 chromosomes in our egg, then when we have our baby, we end up with our expected 46 chromosomes. So we got two primary goals of meiosis, to get us to the right chromosomal number and to create genetic diversity. Those are two primary goals of meiosis. So how do we get there? How do we get the genetic diversity, and how do we get to the 23 chromosomes? Well, the genetic diversity occurs right at the get-go, at the very, very beginning. It occurs right here in prophase 1. And what happens is these chromosomes, if you see how closely they are put together in our visual, they practically hug. And it's almost like a little bit of go fish. They shuffle that DNA. They exchange chunks of their DNA. And we don't really, even though it occurs in prophase 1, we don't really see it in our visual until we get to anaphase 1. We see here how there's orange on the bottom of the purple chromosome, there's purple on the bottom of the orange chromosome, same thing down here. And that's showing us we've got our genetic diversity, and we've got our genetic crossing over what happened from that point forward. We see it in all of them from that point forward, how the purple and the orange has mixed. Okay, so we solved that first problem. We solved that first issue of we want genetic variation. We've got it. Now, how do we get to 23 instead of 46 chromosomes? We do that by 2 divisions right here. So our first division, prophase, condensation of those chromosomes, lining up at the plate, splitting them at anaphase, getting them almost 2 new cells in telophase. But at that point, we've got 46 chromosomes. That's too many. So we go through a second division. The exact same phases, they just add a number 2 on the end of them. And they do generally write it in Roman numerals. So chromosomes are condensed. They line up at the plate. They are split. And then notice, now we have 23 total chromosomes in each gamete, and we're ultimately in cytokinesis going to have 4 separate cells. One right here, 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 and here. Okay? In that way, we've solved our issue of needing genetic diversity, and we have decreased our chromosome number. So let's pull it all together. Mitosis, that is somatic cells, okay? That word somatic really is just another word for body cells. Meiosis, germ cells, the broad term for sex cells. That's all it is. Meiosis starts with 46 chromosomes and ends with 46 chromosomes in 2 identical cells. Meiosis starts with 46 chromosomes and ends with 23 with 4 genetically unique cells. Mitosis goes through 1 division, meiosis goes through 2 divisions. Okay? All right, moving right along here. So our key takeaways. The cell cycle encompasses DNA replication and mitosis. These are important for growth and repair of bodily tissues. DNA replication is the process where the cell doubles its DNA, it duplicates its DNA, it makes identical DNA to the original cell. Then mitosis is that 4-step division process that's dividing our body cells. And the word somatic is a synonym for body cells, everything but sex cells. And this is where one cell divides into 2 genetically identical daughter cells, and it just has 2. Meiosis is the division of germ cells. Okay, those are the sperm and the egg cell precursors. This involves 2 divisions, and it has 4 genetically diverse daughter cells. And they're going to have half the chromosome number. And that's important for that sperm and egg to unite and have the correct number of chromosomes. Okay, we've got some questions. What is the primary purpose of DNA replication? Excuse me. Is it to create genetic diversity in offspring? Is it to produce identical copies of DNA for cell division? Is it C, to repair mutations in the DNA sequence? Or is it D, to convert genetic information into proteins? Get some answers out there for us. I'm seeing some correct answers. Well done, guys. Okay, I think almost everybody got this correct. So very well done. The correct answer is B, to produce identical copies of DNA for cell division. Now let's take a look at the others. Remember that DNA replication is where DNA is being doubled, okay? And it's an identical copy of the DNA. And so A says to create genetic diversity. This is what makes A wrong, okay? Now, if we were thinking in terms of genetic diversity, that would be called crossing over, okay? That would lead to our genetic diversity. C, to repair mutations in the DNA sequence. While our cells do have features that repair mutations in the DNA sequence, that has nothing to do with DNA replication. And then D, to convert genetic information into proteins. That will be our process that we will talk about here momentarily that relates to protein synthesis. Okay, so that's what makes those incorrect. Let's do another. Human cells contain 46 chromosomes. After a cell undergoes mitosis, how many chromosomes will each daughter cell contain? A, 23, B, 46, C, 92, or D, 12? Get some answers out there. Okay, I think almost everybody had this correct. Now remember, this was mitosis, and the correct answer is 46. Okay. Now, sometimes people are like, oh, how do I tell the difference between mitosis and meiosis? Now, guys, I got to tell you, I used to teach high school. So I come up with silly little things to remember. So mitosis, this sounds like toes, and toes are our body cells. And our body cells have to have all the same chromosomes as every other cell in our body. So that would be 46. Now, meiosis, M-E-I-O. I'm writing this with my mouse, so bear with me. Meiosis, remember, that is making the cells for sperm or egg, okay? And remember that we need 23 in a sperm, we need 23 in an egg to make 46. Now, the way that I remember then meiosis is only 23 is that sperm plus egg makes me. Okay, if that helps you, great. If it doesn't, well, maybe it'll help someone else. All right, we are moving on to genetic material and protein. So in this case, we're going to look at the relationships between genes and proteins. We're going to look at the role that the genetic code plays in protein synthesis and how that influences our traits. We're going to look at how we build proteins through this process of transcription and translation. And then we'll also look at mutations when things go wrong. So genes, remember, hold the instructions for making proteins, and it's actually proteins that determine our traits. So proteins are responsible for traits like our eye color, our hair, even our behavior. And it's the genetic code in DNA that is going to designate the order of the amino acids in protein synthesis that determine these traits. So protein synthesis. Now, one thing I want to tell you guys is anytime in science you hear this word or you see this word synthesis, think to make. All this means is we're making a protein, okay? So protein synthesis involves taking the recipe that's stored in DNA and getting it out to the ribosome where the protein is built. And it occurs in two main stages. Transcription, this is going to occur in the nucleus, and this is where the DNA code is being copied into mRNA, and translation in the cytoplasm on the ribosome, where that mRNA is translated into the protein. Okay, that's what's going there. Someone noted that you can also think of synthesis as coming together. That might be one good way to think of it as well. So they're going to take a look at each of these phases next. Oh, but before we do want to make sure we remember a little bit about RNA versus DNA. So remember that RNA is single-stranded, unlike DNA. DNA is double-stranded. Okay, see here are visuals. There's DNA, there's RNA. Remember that RNA is going to have uracil instead of thymine, and that its sugar is ribose instead of deoxyribose. That's where the name comes in. Ribonucleic acid versus deoxyribonucleic acid. Okay, there are three types of RNA involved in protein synthesis. We've got our messenger RNA, our transfer RNA, and our ribosomal RNA, and those are all involved in protein synthesis. Now, the first step, transcription. This is taking place in the nucleus, okay? And in this case, we've got another enzyme, RNA polymerase, that's going to guide the production of mRNA. Now, the way I think of this is that because DNA cannot leave the nucleus, we're essentially making a photocopy of it. And that photocopy is in the form of mRNA, okay? And just like a photocopy is slightly different than an actual copy, our adenine is going to pair with uracil instead of thymine because it's DNA pairing with RNA. And then cytosine and guanine are going to be the same. And now we've got our newly formed mRNA strand, or like I said, it's like our photocopy of the DNA instructions to make the protein. And so once that copy of the DNA in the form of mR is made, it's going to leave the nucleus. It's going to travel into the cytoplasm where it's going to land on a ribosome. And it's on that ribosome where translation occurs. Okay. And so if we look at our visual over here, what we've got is this big green blob is considered, this is our ribosome. I'm just going to write an R since I am using my mouse here. This is our mRNA. So that's like our photocopy of the gene we need to make this protein. And it's laying out on this ribosome. So that's our mRNA. Now, what's going to happen is the ribosomal RNA is just kind of overseeing this process, making sure it's occurring correctly. The big star of the show at translation is tRNA. tRNA is much like our FedEx driver or UPS driver. It is delivering the amino acids because remember, amino acids are the building blocks of the protein. So it's going to read the mRNA strand by linking up the anticodon, which is on the bottom of the tRNA, with the codon on the mRNA. And you might be like, wait a minute, what's a codon? All a codon is a 3-segment section of the mRNA. That's how it reads the mRNA. So much like our UPS driver reads our address in digits and a certain quantity of numbers on our house, the RNA is reading that mRNA in groups of 3 called codons. So you consider the codons kind of like the address. And the anticodon is just linking up, making sure it's correct. And in that case, an A is going to pair with a U, just like we see here in our visual, a U with an A, oops, went too far, and a G with a C. Okay. And when that tRNA realizes it's at the right spot, it drops off an amino acid. That's what we're seeing here. And so those amino acids start linking up. And when that mRNA is completely read, then the polypeptide chain is formed. It's completely formed. And it's going to travel to the endoplasmic reticulum, the Golgi apparatus, wherever it needs to go to finish its three-dimensional shape. Now, next, we're going to take a look at what happens when things go wrong. So a mutation is a change in the DNA nucleotide sequence. So it's a change at the very first stop. It's a change in the recipe book. And it can occur typically during replication when the DNA is making a copy for cell division, but it can also occur due to environmental factors. You know, when we talk about how there are things in the environment that can cause cancer and things like that, they might be causing cancer through the act of mutations. So there are 3 primary examples we want to talk about. The first one is a deletion. And this is just like what it sounds. It's when part of the genetic code is being removed. So it'll be a segment of DNA that just gets pulled out of there for some reason during replication. We could have an insertion. That's where a chunk of the DNA is added. We can also have a substitution. And this is where maybe there was supposed to be an A there, but now there's a T. Or maybe there was supposed to be a G there, and now there's a C. Okay. Now, most of these are harmless, but some can cause health problems. Most of the time, we have repair mechanisms that's going to fix that damaged DNA. But if it escapes these repair processes, this is where we can get cancer or we can get other genetic disorders. All right, we got some key takeaways from this section. Genes determine the formation of proteins, which shape our traits. Protein synthesis includes transcription and translation. If you need to remember which comes first, put them in alphabetical order. Notice that the word transcribe is going to come because of the C before translate, and that will help you remember which comes first. Transcription is where we're making that mRNA strand. Remember, I consider that mRNA like a photocopy. Translation is in the ribosome or in the cytoplasm on the ribosome, where that mRNA copy is being read to form the protein. And this involves mRNA, rRNA, and tRNA to generate that protein. And then remember that a mutation is essentially a mistake or a change in the DNA. And it can impact gene function. It can lead to cancer or genetic diseases, but most of the time it's harmless. All right, we got some questions. Which of the following best describes what occurs during transcription? A, DNA is copied directly into an amino acid sequence. B, RNA polymerase aids in the synthesis of a complementary mRNA strand. C, ribosomes assemble a protein by reading mRNA codons. Or D, transfer RNA delivers amino acids to the ribosomes. Get some answers out there. All right, we're going to talk about this one. I think that protein synthesis can be really challenging. So let's talk about this. But for those of you that did have it correct, our correct answer is B. But let's talk about why it's the correct answer and why the others are not. Okay, so remember that polymerase is an enzyme, okay, and it's helping form that mRNA copy. That's what's happening in that first step of transcription. Now let's talk about why the others are incorrect. A lot of people thought that A was correct, and it's not. Because DNA is not being copied directly into an amino acid sequence because DNA cannot leave the nucleus. That's the problem. If we could take DNA out to the ribosome, then that would probably be just fine. But we can't. We can't pull DNA out of the nucleus. And so we have to have RNA make that copy in order to get those instructions out to the nucleus. One way that I kind of look at it is that DNA is like a recipe book that can't leave the library. And so we make a photocopy in the form of an mRNA strand to bring that recipe home to make our protein. Okay. Now, C, ribosomes assemble a protein by reading mRNA codons. That's translation. And transfer RNA delivers amino acids to the ribosomes. That's also translation. Okay. All right, let's do another. Which of the, what's this? My screen is covered here. Okay, which is the role of tRNA during translation? Is it A, tRNA transcribes DNA into RNA? B, tRNA unwinds the DNA double helix? C, tRNA brings the correct amino acids to the ribosome, matching them to the mRNA codons? Or D, tRNA corrects mutations in the mRNA strand? Let's get some answers out there. We're getting some correct answers. Woohoo! Okay, I think almost everybody got this correct. Our correct answer is that tRNA brings the amino acids to the ribosomes, matching them to the mRNA codons. Very, very correct. Yes. All right, let's talk about each of these. A, tRNA transcribes DNA into RNA. No, that would be mRNA, and that would be going on in the nucleus, and that would be transcription. tRNA unwinds the double helix. No, that's an enzyme that does that. So these completely are, that's relating to transcription. This is relating to an enzyme. Then D, tRNA corrects mutations in the mRNA strand. While our cells do have correcting mechanisms, that is not the role of tRNA. All right, guys, we're doing a nice job staying right on time. We are on our last component of the night, laws of inheritance. So in this one, we are going to look at determining what genotype and phenotype mean, what dominant and recessive alleles mean. We'll look at a monohybrid cross, a Punnett square essentially, and even a dihybrid cross. We're going to take a look at Mendel's laws because Mendel's the one who taught us how to analyze genes via a Punnett square. And then we're also going to consider how things are inherited that even Mendel didn't know anything about. So remember that we have this relationship between genes and proteins. Genes are going to carry the instructions for making the proteins, and it's the proteins that determine our trait. So remember that genes reside on our chromosomes. They might be there or there or there. And that chunk of DNA then is what's determining our traits. Now, Mendel's the guy who studied all of this, and he came up with 3 different laws that are important to us when studying genetics. He tells us that traits are determined by our genes, which we just talked about in our previous slide, but each gene has an alternate form. and these are called alleles. Now, what this means is, let's say we're talking about the coat on a dog, and we're talking about the color of it. And we might have a coat color that's black, and we might have a coat color that's yellow. Let's say we're talking about labs, okay? And so we're saying that the gene is the same basic gene. It'll be in the same location, but they'll just be two different forms. So if this is my chromosome, we might say that the coat color for the dog is right here. And this form might be our black coat, and this form might be our yellow coat. They are in the exact same spot. They're essentially the same gene. They're still coding for coat color, but one's coding for black coat, the other one's coding for yellow coat. And so that's what Mendel was telling us. And then he went on further with this law of dominance to say that one of these alleles, one of them in our visual here, is going to be dominant over the other. And that's what we'll see when that trait is inherited. So we'll only see the recessive trait if there's not a dominant trait there to cover it up. Okay, that's what he told us. And that's what we explore when we look at Punnett squares. But before we dive into exploring with Punnett squares, we've got to take a look at these two words, genotype versus phenotype. Now, genotype is the genes that are responsible for that trait. They're the alleles that are responsible for that trait, the pair of them. And it might be big B, big B, big B, little b, or little b, where big B might stand for that black coat and little b might stand for that yellow coat, okay? Now, when we're talking about the genes or the pair of alleles, use the prefix of the word like we talked about earlier in the webinar to help you. Notice how genotype and genes are very similar. Use that to remind you that that is the pair of alleles. Now, phenotype, notice that pheno is the physical characteristics or traits of the organism that's representing, like the black dog fur coat or the yellow fur coat or hair color or eye color or certain diseases, okay? And let the pH of phenotype and the pH of physical help you remember which is which. Now, when we look at those alleles, we also have three other important vocabulary terms. We've got homozygous dominant. Homo, let that prefix homo, same, okay? Let that same help you remember that it's going to be two alleles that are the same. Let the dominant help you remember that it's going to be the capital letter. So it'll be two capital letters, and we're going to see that dominant trait. Homo, same, homozygous recessive, that's going to be two recessive alleles, and our example could be like two lowercase b's. Now, in Mendelian inheritance, in this inheritance of autosomal dominant or autosomal recessive, we also can have our heterozygous form. Remember, hetero can be different. Let that prefix help you. And in that case, we would see an example such as big B, little b. Now, notice that heterozygous is not, it doesn't follow, it doesn't have a dominant or a recessive after that. And that's because the dominant allele is masking that recessive one. And when we see the physical characteristics or the phenotype, we're only going to see that dominant trait. And so they don't specify dominant or recessive. So Mendel's law of segregation, that's our second law that Mendel indicated. And he noted that each organism is going to have two alleles for that trait, such as F and capital F and lowercase f. And that when we go through meiosis, those are going to separate. So a sperm could get the big F or the lowercase f. A egg could get a big F or a lowercase f, but they're not going to get both. They're only going to get one or the other. They're not going to get both. That way, when the sperm meets the egg, our offspring would then have both combinations of that allele. So when you do a Punnett square, you can determine the probability of offspring getting a trait. So we've got an example here. In humans, freckles, capital F, are dominant, and the absence of freckles, lowercase f, is a recessive trait. It's asking us to perform a monohybrid cross. That means we're looking at one trait. Mono, remember, means one. So we're just looking at one trait. What trait are we looking at? Freckles. And it wants us to do a cross between two heterozygous individuals. Okay, so remember, hetero means different. So that means we've got an individual, mom or dad, and then mom or dad here. Okay, now what this represents, like I was saying, mom or dad, this is a possible sperm. This is a possible sperm. This would be a possible egg from mom. This would be a possible egg from mom. And then we would do our cross. Okay, so let's take a look at what we've got here. So when we do our cross, we pull them in. So bring this F down, bring this F over. Bring this F over, bring this F down. Bring this F down, this F over. Bring the lowercase f down, the lowercase over. And then what we report is we report the genotype. We report the ratios or the percentage of alleles and the phenotype, what the potential offspring could look like. So in this case, we have 1 out of 4 that's homozygous dominant, 2 out of 4 that are heterozygous, and 1 out of 4 that are homozygous recessive. The phenotype then, because heterozygous shows the dominant trait, 1 plus 2 is 3. 3 out of 4 would have freckles. 1 out of 4, no freckles. That's the probability. So we would have a 25% chance of no freckles and a 75% chance of freckles. Now, Mendel's third law that we need to be aware of is the law of independent assortment. And this one can get a little confusing. Mendel said that genes for different traits, so let's say hair color and eye color, are inherited independently of each other. Meaning, just because you might get blonde hair does not mean you're going to get blue eyes. Or just because you get brown hair doesn't mean you're going to get brown eyes. They have no impact on each other. Okay, they don't influence each other. They don't impact each other in any way, shape, or form. And so Mendel said, well, then we can actually do what is referred to as a dihybrid cross. Remember that word, that prefix di means two. This is where we are looking at two different traits. And so we can actually do a 16 square Punnett square that shows all the possible combinations that the offsprings might inherit. And it looks something like this. Okay. Now, this is something that we would dive into deeply in one of our on-demand videos, but I can give you some hints, okay? The biggest hint is that the ratio of two heterozygous parents for both traits, so the parents' allele combination would look like that. The ratio of the phenotypic outcome, what they look like, is always going to be 9, 3, 3, 1. Memorize that. Put that on a flashcard. Do something to remember that. Maybe then you wouldn't have to know a dihybrid cross unless they were asking about something other than two heterozygous parents. Okay. And the whole point, excuse me, of a dihybrid cross is to determine the likelihood of getting the two different traits. So if we were talking about hair color, what would be the likelihood of getting brown hair and brown eyes, blonde hair and blue eyes, that sort of thing? What would be the likelihood of getting those two traits together? Okay. All right. Now we want to talk about non-Mendelian inheritance. So it was the 1800s when Mendel was doing those studies. He did not know that genes could impact our traits in ways other than dominant or recessive. But really, most traits are not dominant or recessive. they have various other ways of helping us, or of being acquired, I guess is the word I want to use. Okay, so the ones we'll focus on tonight are incomplete dominance. That's one way to acquire a trait. Codominance, multiple allele, or X-linked or Y-linked. Sometimes they call this sex-linked, because remember that it is the 23rd pair of chromosomes, the two X's that make us female, or the X and Y that make us male. And that is sex linked traits, because they would sit either on the X or they would sit on the Y. So let's take a look at each of these. So incomplete dominance is a blend of traits that are expressed in the phenotype. So let's think about this for a minute. This picture here, these are snapdragon flowers. And in this case, if I were to cross a white snapdragon with a red snapdragon, instead of getting offspring that are red or white, we actually get pink offspring. Okay, so incomplete dominance, the key word to think about with incomplete dominance is blend. If you mixed white and red paint, you get pink. Okay, that's kind of the key thought process there with incomplete dominance. Now, codominance, this is where both versions of the trait are going to show up in the visual, the physical outcome of the organism. Okay, so I like to think about codominance like co-workers. Both people have to show up to work. What we have here is a checkered chicken. That's what they call that. And that's an example of codominance, because it's not gray. It's not black plus white equals gray, but it is both gray and white feathers showing up. And that is the result of a black chicken crossed with a white chicken in this species. And the offspring then are checkered chickens. Now, multiple allele is when more than a single pair of alleles determine the phenotype. So remember I talked about earlier that on our chromosomes, we would have maybe an allele here and an allele here, and that would determine, let's say, eye color. This is saying that there might be another gene over here, and the alleles on it plus the alleles on this one determine that trait. An example of that can be seen in blood typing. So some of us have A blood type, some of us have B blood type, but some of us have A and B blood type. And that's an example of that, where both the A alleles coming from one chromosome and the B alleles coming from another, in order to give us that blood type. Now, our final one, as I talked about, these are what they refer to as our sex-linked genes. And this is when the trait is carried on a sex chromosome. And what happens here, which is really interesting, is that men kind of get the short end of the stick here when we're talking about the X-linked trait. Because if you're female, you get an X chromosome, you get an X chromosome. If you're male, you get an X and a Y. Well, where the guy gets the short end of the stick is if the trait on his X chromosome is a recessive, he doesn't have another chromosome to cover it. So female, oh, she's getting, another chromosome to cover that up. If she gets a capital R on one, she's good, let's say. Where he's only getting one because that trait's not on the Y chromosome. If he gets the little R, well, now he's getting whatever recessive condition is on that X-linked chromosome. And the most common example of that is color blindness. So if you're male and in this webinar and cannot see the numbers in there, that means you've got a recessive allele on your X chromosome. There are very few traits linked to the Y chromosome, because then females wouldn't get them, but it's not completely unheard of. All right, we got some key takeaways. Genotype refers to the combination of alleles, like big F, little f. Phenotype is referring to those observable traits. Remember, phenotype, physical traits. A single dominant allele will mask a recessive allele if we're talking about Mendel's law of inheritance. People are asking about the notes. Yes, you will get the notes as soon as we're done here. Punnett squares are going to determine the probability of inheriting certain traits, and you should definitely get them by 7 to support your request. Most traits, though, are not inherited Mendelianly or autosomally dominant or recessive. They can be inherited via incomplete inheritance, which is a blend, codominant, where they both show up, or where more than one allele pair determines that phenotype. And then remember that sex-linked or X-linked or Y-linked can be on those sex chromosomes. All right, we have 2 final questions, and then I will be happy to share those notes with everyone. Our first of our last question, which of the following best describes the difference between genotype and phenotype? A, genotype refers to the observable characteristics, while phenotype refers to the genetic makeup. B, genotype is the genetic makeup of an organism, while phenotype refers to the observable traits. C, genotype and phenotype are both related to an organism's observable traits. Or D, genotype refers to inherited traits, while phenotype refers to acquired traits. Get some answers out there. All right, I think almost everybody got this one. Well done. B, genotype is the genetic makeup, while phenotype refers to those observable traits. Remember, genetic, phenotype, and that would be physical or observable traits. All right, why is A incorrect? Because it has it at the complete and total opposite, okay? And then C, no, because genotype is not observable traits. And acquired traits are what you get in your lifetime. Like if you were to color your hair blonde when it's not blonde, that's an acquired trait, and that does not get passed on to offspring. Okay? All right, we have one more, our final one of the night. And Mendelian genetics, why do the genotypes big B, and big B, little b result in the same phenotype? Is it A, because in Mendelian genetics, it only takes one dominant allele to mask the effect of the other? B, because big B, little b is recessive and doesn't influence the phenotype when paired with big B? C, because both big B and big B, little b contain only dominant alleles? Or D, because the phenotype is determined solely by environmental factors, not genotype? Get some answers out there. Okay, so we were kind of varied on this one. Our correct answer is A. It only is going to take one dominant allele to mask the effect of the other. Let's take a look at why the others are wrong. Because big B, little b is recessive? No, we've got a dominant allele right there. And doesn't influence the phenotype when paired with big B? No, we've got dominant alleles. C, because both big B and big B, little b contain only dominant alleles? No, that's a recessive allele right there. Or D, because the phenotype is determined solely by environmental factors, not genotype. And we know that's not true. Okay, that's completely silly. That one's just completely untrue. All right, so wrapping up tonight, just remember that we've got our QR codes here. So when I share the slides, you will have access to our free live webinars. You can rewatch tonight's webinars and old webinars. You can access the question bank. You can order on-demand videos, schedule tutoring, or you can look at some of our packages that are put together and give you a little bit of a discount. And that's what the SurePass versus SurePass Pro. The big difference here is you get tutoring with the SurePass Pro. And then remember that you can find us on the socials, on Insta, on TikTok, on Facebook, and you can always email at freenursing@archerreview.com. And then finally, I'm Shelley Harshberger. I was your science person tonight. If you would like to have me as a tutor, you can scan that QR code. You've got Tammy for English and reading, and you got Brandon for math. Okay, to wrap up tonight, we do have a QR code that you can use to give me some feedback. Please do so. I would greatly appreciate knowing how I did tonight for you and what I can do to better serve you in the future. And someone's asking to see the first slide. I can do that for you. But let's keep this webinar feedback survey up for just a minute. And at this point, I am going to put in all of our socials the slides, and you would be able to see that slide then as well. So give me a quick second. I'm going to get those notes out to you while you guys are doing our webinar feedback survey. The slides are out there on Zoom, and the slides are out there on our other social media sites, YouTube, Facebook. Thank you guys so much for being here tonight, and please do take care and take that survey. I'd love to hear from you. I'm going to leave this up for about 1 more minute, and then I'm going to close it out. I did have a request for the first slide, but I am uncertain if you mean the first slide of the entire webinar or if you mean the first slide with all the QR codes. And you will have gotten that in my PDF I sent you. So unless I hear from you, I'm just going to leave it on our webinar survey for the moment, and I'll keep this queued up for about another minute. For those of you that are all set, have a great night. All right, folks, we are all set here. You guys have a lovely evening. Thanks for coming. I'll look forward to seeing you with some future webinars. Take care.